Today, we will cover drug resistance and how to find mechanism of drug resistance in cancer. So Lapatinib, when it was first introduced and FDA approved, many said we have found cure to cancer. And indeed, in HER2 positive breast cancer, Lapatinib show amazing response. In many patients, the tumor completely disappear after a few weeks of treatment. However, after a few months, tumor reoccur, and this time the tumor were more aggressive and resistant to any kind of treatment. We now understand mechanism of resistance to Lapatinib. Lapatinib inhibit HER2, and the inhibition of HER2 could be completely compensated by upregulation of different tyrosine kinases shown here. So in the many resistance tumor, the receptor tyrosine kinases are upregulated, which in turn can activate the downstream genes of HER2 and make the cancer cell independent of HER2. So let's go and talk in detail resistance mechanism of two targeted therapy. First, in prostate cancer, androgen receptor, which is a transcription factor, is an important gene. It is activated by hormone, and it is essential in prostate cancer. So a primary treatment for prostate cancer is blocking the androgen receptor. And let's try to understand how does resistance emerge to this blocking. One of the mechanism is the tumor gain mutation in ligand binding domain, and it becomes independent to the hormone for androgen activation and androgen receptor activation. Another way of the resistance is ERG activation. It's a pioneer factor which increases the chromatin accessibility of DNA. So even the small amount of transcription factor androgen receptor could still activate its downstream gene because the chromatin is already accessible. The GR, which is a very similar receptor and transcription factor as AR, they have a similar motif. So when AR is inhibited, GR can get overexpressed and it can compensate for AR. And finally, the tumor cell can transition from a state where it is not so much dependent on androgen receptor. So one of the case is the tumor can transition from more epithelial to more mesenchymal state, or from more luminal to more basal state, where it is not dependent on androgen receptor. The second targeted therapy we'll talk about is in non-small cell lung cancer, where the mutant EGFR mutant tumors are treated with EGFR inhibitor, because the mutation in EGFR are usually activating mutation. One of the mechanism, like previous targeted therapy, is the binding domain of EGFR gain mutation, and the inhibitor become inactive. It cannot function properly in presence of this mutation. MET amplification, which is another tyrosine kinase. So as you can see here, MET falls in the parallel path to EGFR. It has the same downstream gene PI3K. So if EGFR is inhibited, the activation of MET can activate the PI3K kinase and leads to growth and metastasis without the need of EGFR. Growth factor can also lead to growth and metastasis without the need of EGFR. PI3K, which is downstream gene, can gain mutation by which it can be activated by itself without the need of EGFR. And finally, the tumor can go from non-small lung cancer to small cell lung cancer, where it is not so much dependent on EGFR activation. So if you compare this resistance mechanism in these two targeted therapy, you see a lot of similarity. Many of them are binding domain mutation. There are downstream gene mutation or the receptor tyrosine kinase activation, or the tumor cell can go into a stage. It can switch into the state where they are no longer dependent on the genes which is inhibited by drug. So another key question is whether the tumor already have cancer cell before treatment, which are resistant to the treatment, right? Or during the course of treatment, after treatment, cancer cell evolve and gain mutation so that it become resistant to treatment. So remember, the point mutation rate is quite small, and only the minority of resistance clone gets selected for colon. So to say more concretely, which is shown in this figure, remember the tumor is usually very heterogeneous. The tumor cell, the cancer cell within the tumor, they have different mutation, they have different vulnerabilities. So when the drug treatment is applied, only a small subset survive and they expand. And this is more plausible explanation of emergence of resistance. That the cancer cells were already present before the treatment, and only those cells survive, and then the tumor relapse. Okay. So, what are the ways to find resistance mechanism? Usually, people use cell line model because they are easiest to do a large-scale screen. However, there is a debate whether it's a good model to represent the human system. So what people have already done in thousands of cell lines, they have done the drug screen for a few hundred drugs. So these are the two big cell line consortium which have conducted a drug screen. And from analyzing this drug screen database, we can already identify some resistance. First of all, let's take the example of EGFR. EGFR as a gene name. So cell line with EGFR mutations are more sensitive to the EGFR inhibitor, and this we can identify from this drug screen database. In a similar manner, we can also identify some other gene B whose expression or mutation influence cancer response to EGFR inhibition. And most likely this gene B is then a driver or a mediator of resistance to EGFR inhibition. Right? And if the gene B happen to be druggable, then basically you can combine the inhibition of gene B with EGFR, and that treatment is likely to be more effective. Another way to identify the resistance mechanism is through conducting CRISPR screen or shRNA screen. So basically treat cells with or without drug. And the key question there we are answering is whether knockdown or knockout of gene makes cell more or less sensitive. Right? So if a gene knockdown makes cell more resistant or less sensitive, then that is a viable resistance mechanism. So we will look into this more deeper, how we can identify such resistance mechanism in an in silico manner. So let's revisit this most familiar kind of genetic interaction called synthetic lethality. By definition, in synthetic lethality, it's a symmetric relationship between two genes, where individual knockdown is not lethal to the cell. Cell is happy. But if you knock down the two genes simultaneously, it leads to the cell death. In the area of the resistance to targeted therapy, there is another kind of genetic interaction which we have recently identified, which is called synthetic rescue interaction. It's an asymmetric relationship between two genes, uh, where the knockdown of first gene causes the cell death. But the secondary knockdown of the second gene, which we call rescuer gene, makes the cell happy, cell survives again. And so basically it rescues the cell from knockdown of vulnerable gene, right? One can also imagine another kind of synthetic rescue interaction where rescue happen due to upregulation of the rescuer gene. And this is more likely to be common as an example of EGFR and MET, where knockdown of EGFR was lethal to the cell until unless the MET got upregulated. And this is what we call a DU kind of synthetic rescue interaction. Okay, so how can we identify such interaction? What we can do is we can harness the property of synthetic rescue interaction to identify. And for simplicity, let's just talk about the DU kind, where the upregulation of the rescuer gene causes resistance or rescue. Okay. So one of the key property of synthetic rescue interaction is the cell lines. If the cell lines have already upregulation of a rescuer gene, the gene knockdown of the vulnerable gene is less likely to be effective, right? Because it's already rescued. So that's the in vitro property of synthetic rescue interaction. One can also look at the cancer evolution, the cell evolution implication of synthetic rescue interaction. So if the cell have already a downregulation, or if have already lost a vulnerable gene, it would only survive when it have upregulation of its rescuer gene. So we can look for enrichment of such rescued state in a tumor population or within the tumor, right? And the third thing is if a tumor which have lost a vulnerable gene, if they are rescued, since this is an indication of emergence of resistance, if the patient has such tumor, they are likely to have very poor survival. And then the phylogenetic property just implies that they are functionally similar. So now you can combine all these four property, and then you can narrow down the synthetic lethal interaction. And these are the synthetic lethal interaction which has been identified by applying this algorithm, where the red genes are the vulnerable genes and the green genes are the rescuer genes. Okay. So what can we do if we know the synthetic rescue interaction? So first of all, synthetic rescue interaction determine the patient survival. If you compare among the cancer cohort, the patient who have many rescued events where the vulnerable genes are knocked down but the rescuer genes are upregulated, compared with the patient who doesn't have any such rescue event, there is a significant survival difference. Specifically, the patients which have many such rescued events, they have very poor survival. And if we concentrate on a subset of genes which are target of specific drug, then we can identify mechanism of response and resistance. So for example, this antitubulin drug, it has this bunch of 3 rescuers. And now if we divide the patient who are treated with this antitubulin drug, the patient who have overexpression of this rescuer are less likely to respond. And that's what we see here. This is the response rate, or just a fraction of the number of the expression of the rescuer gene between responder and non-responder. So just by looking at the expression of the rescuer gene, we can determine whether the patient would respond to therapy or not respond to therapy. And finally, SR can help you to create or to devise ideal drug combination. So here is an example, which is DNMT1, which is a vulnerable gene, and it have bunch of DU rescuer and bunch of DD rescuer. So the upregulation of this set of genes uh, is likely to rescue the inhibition of DNMT1, while the downregulation or inhibition of these two genes are likely to cause rescue to DNMT1 inhibition. And accordingly, if you think about if you co-inhibit DNMT1 and its DU rescuers, then those two drugs are likely to be synergistic. And if you inhibit the drug, co-inhibit their DD rescuers partner, then they are likely to be antagonistic, because if you inhibit the DD partner, it is going to cause resistance. And accordingly, we actually found that 71% of the predicted DU rescuers were synergistic, while 64 were antagonistic. And we didn't find any of the SRDU rescuers were antagonistic or vice versa. So in summary, resistance to target therapy is prevalent, and resistance mutation are already present in pretreatment tumor. Combination therapy is way to go to mitigate the chance of resistance. And synthetic rescue interactions identify the molecular pathways important in resistance and help us to devise effective combination therapy so that we can mitigate the resistance. Thank you.